So we finished with the light reactions. They're called light-dependent reactions because you have to have light in order for them to work. Photosystems, light reactions, all kinds of things that can be called. So let's just real quickly remember what we ended up with when we were done. So we had electrons that came out of water, and they were excited and given to NADP, which was reduced to NADPH when it was carrying high energy electrons. So we've got those. That's what glucose has to have. And we've got ATP, which is also required because we're going to be taking lower energy, more stable precursor molecules and rearranging them so that conformational change requires energy and they become less stable and weaker, but that's what glucose is made out of. So we've got to have ATP for the conformational changes, electrons for the bonds, and notice where that all of that was made. Made in the stroma, which is where the Calvin cycle is going to be, is where the Calvin cycle occurs. So that's what we're doing next. So this would be, they could call it the light in independent reactions because Calvin uses the products from the light reactions, but actually light energy doing anything in Calvin does not happen. So most of the time people are just going to call this the Calvin cycle. And because the Calvin cycle's job is to assemble carbon dioxide with carbon dioxide with carbon dioxide, so you end up with glucose at the end, I write this like that to help me remember. These two C's together remind me, oh yeah, that's right. Calvin's job is to put carbons together. You know, Calvin, cycle. Okay, Calvin cycle. So that's its job. So we're going to simplify something that is, I mean, it makes Krebs look like kindergarten. Calvin cycle is extremely complex. So the best way to think about it is to think about it, because it is a cycle, and we're going to draw this, obviously, as a circle. Just to remind myself that it's a cycle. And it's going to have three main parts. So there's three lumped together common stages within it. So I'm just going to make it into a pie. And then me being me, like I want them in different colors. So pause, and I'm going to add yellow here, orange, and then green. I'll be back. Okay, so now I've got everything ready, and I'm going to have my three different stages. And I'm just going to come over here and I'm going to number them. So the first one is called fixation. I'm pretty sure most people anymore don't know what a fixative is. Like Hunter does, because he's an art major and he knows all about fixatives. Do you know what fixative is? You heard the word? So fixation is the process of fixing. Now, I'm not fixing as in repairing. It's the same thing as a fixative. So I looked it up so that you could see it's the process of making something fixed. Well, that didn't help. Fixative, fixed. You can't use the word in the definition. Or permanent. So a fixative is like glue. So if you've ever seen the commercial for Fixodent, it's the process of making your dentures stuck in your mouth. So it's making something attached pretty much permanently to something else. Now, I'm pretty sure Fixodent doesn't keep your teeth locked forever in your mouth. But, so this word fixation, what they're saying is we are gluing or permanently putting together two things. That's what this process is. So it is the process of fixing. Fixing, not repairing. Fixing as in sticking, putting together. Then, secondly, you have reduction. So this word now should be common to us. This is where the electrons are going to come in, because something will be reduced by gaining electrons. The last step, because it's a cycle, is you've got to remake whatever you started with. You've got to regenerate that initial precursor beginning molecule. So this is called regeneration. So fixation, this is driving me crazy, is the process of taking this precursor molecule, this beginning molecule, and attaching, fixing, sticking something to it. So the Calvin cycle starts with this really cool molecule, and we're just going to call it RuBP, ribulose bisphosphate. So it's R little u big B big P, RuBP. It is the beginning and the end of the Calvin cycle. It's a 5-carbon, I might as well put that over there. It's a 5-carbon molecule, and it's what we start with, what we fix, attach something to. You can guess what that's going to be, because what do we use to make a carbon-based molecule? Well, it'd be carbon, and the form we use is carbon dioxide. So we're going to fix carbon dioxide which is 1 carbon, to this RuBP. There's an attachment there that's called fixation. Then what we're going to do is, now this is way simplified. Okay, I just don't want to keep saying that. It looks super simple, like, man, you got 6 carbons. It does not remotely add up like that. The numbers are bizarre. Like at one stage you go from having 5 3-carbon molecules, which, okay, that's 15 carbons, I can multiply, to 3 5-carbon molecules. Oh, okay, that's still 15 carbons. So this is not simple stuff here. So now that you've got this carbon dioxide that's been attached to this precursor RuBP, and we'll come back to the, we might as well write it on here. The name of the enzyme that does that is a really weird name. It's called Rubisco. Let me put it in a box. Which, its name is a combination of the crazy long name that it's made out of. But it's very old, highly conserved enzyme. It's probably one of the most common proteins on Earth, because enzymes are proteins. It's just unique. We'll talk a little bit about some of the things that it does, but its main job is to add, fix carbon dioxide to RuBP. That's its job. So that's Rubisco. Rubisco's an enzyme. Its job is to put these two together, carbon dioxide with your precursor RuBP. So that's the whole process of fixation. A whole lot of yellow for a whole little bunch of stuff. Reduction, that tells me that I'm going to be adding electrons. So we're going to now use those products from the light reactions, the ATP and the electrons in the form of NADPH. So we're going to have ATP that's going to be used here, in this reduction part. ATP is about conformational change. So if you kind of think of the parts of a molecule, the atoms in there, as being Legos, it's rearranging the Legos. And the less stable bonds are less stable because they're more energetic. So that's why we use the ATP. So this ATP now is for conformational change. And then we're going to also use, oh my goodness, the electrons that are being carried by NADP in its reduced state of NADPH. So we're now going to take this RuBP plus the carbon dioxide, rearrange it, make some bonds, and what you end up with is the molecule that is generated by the Calvin cycle, which is glyceraldehyde 3-phosphate. Sometimes you'll see it called PGAL, but you're going to get out G3P. Now, as I don't want to get into this, I just want to say, how do I even describe it? This is not done in singular stuff. So you end up with like a bunch of RuBP added to a bunch of carbon dioxide. So you really have a bunch of carbons, and you do this hocus pocus, rearrange them, and you end up with, after you get through here with all of this, 6 of these glyceraldehyde 3-phosphate. Okay? You end up with 6 of them. You leave 5 of them in the system, in the cycle, and you let 1 out that you use. So the remaining G3P that you, like this is the one, like it's, you know, if you're doing something and somebody says, hey, can I have some? If you have 6 of something and you give them 1, okay, you gave them something. So you give 1 out to be used. The other ones stay in this cycle, and G3P, glyceraldehyde 3-phosphate, is going to go through a conformational change to something called triose. Tri means 3, ose is sugar, triose phosphate, called TP. And then, hocus pocus, a bunch of changes. Oh, you mean we're going to rearrange some things? Wouldn't we need ATP for that? Yes, you would. So you'll use some more ATP, and you'll end up back with the RuBP that you started with. I think sometimes when we make things simple, it's kind of crazy. So you start with this 5-carbon molecule RuBP, and the first step is to bring carbon dioxide from the atmosphere into this cycle. Let Rubisco combine or fix the two. So what you've done here by bringing in carbon dioxide is you've increased the number of carbons in this whole cycle. Then you're going to rearrange them, make some bonds, and they're going to be rearranged into the format of glyceraldehyde 3-phosphate. So if we think about it, we can simplify it this way. Glyceraldehyde 3-phosphate has 3 carbons. It would take 3 carbon dioxides coming in, right? So we have a certain number already in the cycle. We bring 3 new ones in. We're going to hocus pocus rearrange them all. We let those same, not necessarily those same, but we let 3 carbon dioxides, here we go out, except we've put them together into a glyceraldehyde 3-phosphate, which is like the Swiss Army knife. We can make anything from it. Then we have another set of, hey, it's a cycle. Shouldn't you go back to what you started with? Oh yeah, my bad. Glyceraldehyde 3-phosphate into triose phosphate into RuBP again. And then we can continue through again. So let's add a couple of details here. If we are going to use ATP, then what will come out is ADP, right? We can go ahead and put the inorganic phosphate, but whatever, because that's what happens. This is my full ATP. My empty is ADP. Same thing here. So as I use ATP, ADP is created. You know where I'm going. Once I pull the electrons out of NADPH, it's the full. Then I'm going to create the empty, the oxidized form. I create an ADP. All of the empties can go back to the light reaction, which once we start writing the stuff down at the bottom, then we'll put all that information in there. Okay, so that literally is the Calvin cycle. When you're done, you end up with this. Let's make this important. Blah, 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 blah. I feel like I'm drawing a sheep. So this is the outcome molecule of the Calvin cycle, glyceraldehyde 3-phosphate. And then when we get down here and we talk more about this, right now we're just drawing, we'll talk about what you can make from these molecules that the cell, the animal, not animal, that the plant cell could use. So we'll come back to that. All right? So are you okay? I hope you're okay because we're done. That's the end of photosynthesis. We'll just come back later now that we've drawn. We'll come back and add the notes.